This is a production of Cornell University.
So thank you, Margaret, for that wonderful introduction. At one moment, I kind of almost cried because I was like, oh, gosh, I am going. Kind of switching gears, I guess, because like you mentioned, we kind of have limits on our time here. But obviously the topic's up there at the moment. But so basically, the Green Revolution. The Green Revolution increased yields in the crops that we eat, and it selected down to very few crops globally. And one of the costs of taking that focus on a few crops and focusing on yield in particular meant there was actually a reduction in the concentration of the mineral elements that we have in many of the foods that we eat, and a reduction in diversity in some of the many of the foods that people eat as well. So with that as well, there's an increasing interest in the nutrition of the food that we eat as well. So understanding interactions between genetic and environmental factors is paramount to understanding nutritional quality of the food that we eat. Oh, what do I do? Okay. So there are various organizations that provide information, such as the USDA and the World Health Organization, that will provide information on the types and diversity of foods that we should in order to attain the essential and beneficial elements that we require in our diet. So 22 mineral elements. And so these if we have any reduction in this diversity of food, we're expected to have a reduction in the amount of mineral elements that we have in our diet as well, which would affect our health. So you may be familiar with this, and this is from the USDA, giving an idea of the types and the quantities of food that we should eat in our diet in order to get these mineral elements that we require. But as we're aware, all around us, we don't have to look too far to see that there are some people is this too loud? Maybe a little too loud? There may be some people that actually don't get the types of diet that they require in order to get these minerals in their diet. So let's look at Baltimore. And in Baltimore, there's a map here, and we can see that the red here represents food deserts. And a food desert is defined by a lack of one or more of these three layers here. So the first one here is physical access to healthy food, affordable healthy food, and desire and knowledge to eat healthily. So if we were to have any one of those lacking in the area, geographical location that we live in, we may expect that there would be the possibility we'd have a reduction in the healthy food that we can access, and the diversity of mineral elements that we may have in our diet as well, and therefore affecting our health as well. So, but that also happens on a global scale as well. So here we have a map of the world, and this is the incidence of global zinc, iron, vitamin A, and iodine deficiency in the world. And just looking at that, quick brief look at that, it shows you that there are many countries around the world with, from mild to alarmingly high incidence of deficiency of these various elements. But more than that, considering just these elements here, maybe one or more of these elements, about 2 billion people globally are expected to be deficient in one or more of these elements. So it's a really important question here. So, but it's easy to resolve. So here, just supplementation is one of the easy ways that we could actually just remove this problem for people. So supplementation directly with tablets is iron, for example, or supplementation to the plant in fertilizer form. We could have foliar feeds and that type of thing. Or another way may be fortification of processed foods. But we actually argue that biofortification, so that's using the innate genetics of the plant is a sustainable and long-term method approach to actually providing the right nutrients to people that actually don't have access to the food in the right way that they should do. Actual delivery of nutrients is potential, especially to those people, the marginalized poor. When we consider people in sub-Saharan Africa, for example, for places that around the world that actually don't have as much access to the mineral elements that they might require for a healthy diet. So the question then is if we're going to do that, what is it that we need to know in order to address this question? So from research in plants, we become aware that we're aware that genetics regulates the elements that go into the plants that we work with, in the crops that we work with. But we're also aware that one of the other factors of that is the soil type, the soil pH, fertilization, and these other factors as well. So genetics here we've denoted as uppercase G, and environment is denoted as uppercase E. Sound familiar, guys? I think so, yeah. But then there's another element to this as well. It's the interaction of the both, G by E, okay? So before people start throwing things at me, I'm going to keep moving. So, okay, so I want to briefly talk about some of the genetics behind that. And I'm going to just talk about iron now. So the path of iron through the plant. So I'll do it from this one. So first we look in the rhizosphere, and here we can see insoluble iron within the rhizosphere, which is being chelated and then passed through channels. And I've highlighted this one channel here, yellow stripe one, because we're going to talk about that a little bit later. So from the yellow stripe one, we're going to take this path that takes through the cellular and intracellular pathways, going past the Casparian strip, and then towards the xylem and the phloem. In the xylem, it will be taken up through the plant and into places such as the leaves. And then in the leaves, because we're dealing with iron now, which can be, we need it as an essential element, but it's also toxic in high levels, what we can do is, what it does is that something like the N ramp here will actually take that iron that's in too high quantities and actually put that and store it in the vacuoles and keep it safe from the other functions in the plant. And then continuing this passage up to the parts of the plant that we eat, then we find that the iron will be in those areas there. So there's this whole process that really, we know parts of it. I mean, I've just explained it to you, right? We know parts of this process, but there's also a lot that we don't know. So that's what we want to talk about today. What don't we know? So maybe let's talk a little bit about the ionome. So the mineral, nutrient, and trace element composition of an organism. So that's the definition of what an ionome is. But it comes from, it's like an intermediate of two other terms as well. So the first one, sorry about this, it seems there was an incompatibility with the computers, but so the metallome, I have to say that correctly, that's the inorganic metals within the plant, which is smaller than the ionome. And then we have the organic low weight molecules and inorganic metals, and that's the metabolome that maybe we're more familiar with. So then the ionome then is the nutritionally relevant minerals and trace elements. So now that we know that, so how can we influence the mechanisms to improve nutritional quality? How is it we can really get in there and understand the genetics in order to make it work for what we want? So the problem is currently known genes do not explain the whole story of elemental homeostasis. So our solution on that, our take, is we want to know the complete set. How is it we get to know more of what's going on, the complete set, to give a bigger part of the story? And so our method is using association mapping on 20 elements that, and these 20, remember there was 22 elements for the, that we were looking at essential elements earlier. So we're looking at 20 of those, though, on 10 diverse environments and 4 different populations. So that's the complete thing that we're looking at there. But I'm going to talk to you about just 3 of these populations. And you may be looking at this going, what on earth is that? So I'm going to just walk you through it. So these squares represent, on the triangle, they represent the genetic diversity that we find in each of these populations. So this one over here is probably quite familiar to most of you, is the nested association mapping panel. And the comments that I have in the middle are kind of relative to the other populations. So here we have the NAM is expected to have greater power than maybe the second maize diversity panel. Oops, sorry. And then great resolution and less diversity. The maize diversity panel, the second one here, is expected to have less power, statistical power, and greater resolution and greater diversity. Now, you might be wondering why this square is in the middle here. And that's because some of the recombinant inbred lines that we find in the NAM are actually also found in the diversity panel as well. Then coming to the third population over here, the heterogeneous inbred families, we have the square again. And that tells us that these HIFs are actually created from the NAM. And these HIFs that we create are nearly isogenic lines, which is a slightly different question than we were asking for the first two populations. So I'm going to talk just very briefly about the NAM. I'll be quick, don't honestly. 25 NAM founders, so of reasonable diversity, genetic diversity. These 25 founder lines were all crossed with B73. They were selfed to the F6 generation. And then we have 25 recombinant inbred families. And within each family, we have 200 recombinant inbred lines, a total of 5,000 individuals. And those would also be found in the diversity panel as well. So, okay, so we're getting into some of the data now. So, working in the NAM. So, in the NAM, we planted at 4 different locations, quite diverse locations. And then in these locations, we're looking at elemental accumulation by location. So here I've picked 2 random elements for you. One is manganese and one is molybdenum. Now, the concentrations at each location are seen in the box plots. And then below that, on the x-axis, you can see the locations there, but below that again, we have the pH, the soil pH for each of those locations. And just to try and make life a little bit easier for you, I've colored those by the amount of accumulation of the availability of that element that we would expect with a pH of that value. So for example, in North Carolina, we have a pH of 4.5. We would expect for manganese to have a low availability. Likewise, in North Carolina, for molybdenum, we would expect to have a poor availability again once there. But what do we see? The data that we actually see is that the concentrations for each of these within the kernel that we're taking here is actually, we expect to have poor availability, but we don't have the lowest for the kernel concentration. And likewise, for the manganese, we don't have the lowest again. So that tells us that, you know, the concentrations in the kernel are not tracking directly with the pH that we see in the locations that we're looking at. So that tells us, once again, there's something else at play here. So, well, I get to the point here. The point is, we believe it's the genetics that's at the greater proportion of this that's spelling out the story for you. So when we look at the heritability, broad sense heritability, which is the genetic contribution to the phenotype that we're observing, when we look at these 2 at the bottom here, manganese and molybdenum, we can see that their heritabilities are 0.6, which is a reasonably high heritability. But then let's look over to these other ones here, and we find that we have some others here, 0.3 or lower heritability, which tells us that actually there's probably a greater influence of the environment on that phenotype as well. So let's take that point home there. So we're talking about the G and the E, and the G by E together. Yeah. So the next slide. So, okay, great, right? You're like, okay, what's going on here? Okay, so now what I want you to do is I want to just take you on a little journey through the process of preparing the data for association analysis. And so I'm just going to sort of talk briefly about these, but get used to these slides because these slides will be coming up again. And so I don't have to talk through them again. I'm just going to give you the same type of thing. So here we have the ICP-MS. So what that stands for is inductively coupled plasma mass spectrometry. And what that is it will actually record the concentration of the element that we're looking at and give us some raw data and tell us how much of that is actually in the plant. And then for that, we sampled, for the NAM, roughly 50,000 seeds. Then here, we remove outliers using the median absolute deviation, 2.5 standard deviations from the median. And then here, we do a Box-Cox transformation. And then here, we do the best linear unbiased predictors. And that's basically to correct for errors in the data that we get for each line. And we did those for element for a line. Okay, so then we moved on from here and we looked at joint linkage, which is considered to be a more conservative method of doing this analysis. And then below that, we did association mapping, genome-wide association study. And then for resolution, within the genome, we did approximately 29 million SNPs. So here I put QTL-like rain, because literally that's what it's going to be like. We get so many QTL, they're bursting out of your ears. And so that's where filtering comes in. It's really important. To get the correct significance of the threshold for filtering allows us to filter between the sunny QTL, the good ones, and the thunderous QTL, the bad ones. And so, so that we process that and take that data and then, so let's have a look at some of that data. So for our joint linkage results, and I'm not going to bore you with too many details here, but what we have here is individual locations, so North Carolina, New York, Puerto Rico, and Florida. And then these ones would be a QTL that's found at multiple locations, so 2 or more locations. And then right at the bottom here, so this is a subset of all of the 20 that we were looking at, but this is all of them added together. We find that the numbers that we get for individual locations are far higher than the numbers we get for multiple locations. And that also plays into the whole G by E idea as well, that actually some QTL, they don't explain everything across every location. It's just some things only explain specific things within one location. And let's look at the GWAS as well. And this is, you know how I said it was more liberal? So we're getting thousands here. But then look at 2 or more locations, 29. So that makes you feel a little deflated after that, right? Anyway, so that's the results that we get for the joint linkage and the GWAS. And let's have a look at them a little bit as well. So what we have now is, this is the joint linkage and GWAS hits for manganese, and everything is overlapping now. So but we had all these thousands of QTL, but now what we have is not just thousands, but they're all stacking up upon one another. So they're actually starting to make sense a little bit. They're telling us a story. And so this is for manganese, and this one's for molybdenum. And we were getting so many thousands of QTL that we were like, well, what are we going to do with this? How are we going to sort of tell a story? So the PI that we're working with, Ivan Baxter, so his lab actually felt a good strategy to do would be to create some software. So this is ZBrowse that we're looking at now. And it's a browser that you can use to actually visualize many of these QTL. Anyway, so the next one is phosphorus. And you see how they're stacking up nicely there. So we can actually really sort of look down at the region of the chromosome that's of interest. So let's have a look at some of the genes that are under some of these stacks. So we have just a random selection here. So phosphorus, manganese, and molybdenum on chromosomes 9 and 1. And here, once again, so we have the sulfate transporter. And then, what was I was telling you about earlier? The N-ramp. So here we found the N-ramp. So we know this story already. And then we have the MOT1. So the reason I'm pulling these ones out for you is because we know these stories already. So I can say to you, oh, well, we've got what we expected. But actually, there's a lot we don't know as well. So many of these thousands of QTL that we got, they tell other stories that maybe we didn't expect at all. Yeah, so there's more stories to tell. But I want to talk about this one here a little bit first. So MOT1. Now, MOT1 is one of our favorites because it was originally found in Arabidopsis in the mitochondrial envelope, and that was by the PI, so I gotta like it, I guess, Ivan Baxter. But also it was discovered by Josh Cobb as well, so from the McCooch lab in 2013. And then the reason that we like it is because MOT1 is a key regulator of molybdenum in the genome, and it explains about 30 to 70% of the variants that we see. So successful detection of MOT1 is used as a quality check for the GWAS that we do. And because it's so almost a qualitative gene that we're able to really, if we don't see that gene showing up in our results, then we start to worry a little bit. So now that we've looked at the NAM, we need to think a little bit, what do we learn from this? So what adaptations needed to improve analysis? So it's necessary to increase, we've learned that it's necessary to increase statistical power in our association analyses, and then also to increase the resolution as well. So, look familiar? So we're back here again, yeah? So now let's look at the maize diversity panel. And that's this big one in the middle here. So we're expecting to have less power, great resolution, and great diversity. So we planted these in Florida, Indiana, Missouri, New York, North Carolina, and Puerto Rico. And that was performed, we're talking about increasing our resolution, that was performed on approximately 83 million SNPs, before filtration. And then we implemented a multi-locus mixed model, and that was in order to reduce false discovery and increase power as well. Okay, so we're back to this again, and we're looking at the concentrations at locations of iron and molybdenum. Here are our locations. And then the green here is where we would expect to see a good availability of iron. But when we look up here, we can clearly see that we're not getting that. In fact, it's the lowest of all of them. When we look here for North Carolina again at 4.5, okay, it's maybe one of the lowest, but what about explain for Florida as well? Because that's one of the lowest as well. So clearly something else is going on here. Oh, sorry. So when we look at the genetic contribution to the phenotypes that we observe, we see that in North Carolina, New York, and Indiana, for iron and molybdenum, within locations, consistently we're getting higher results for the genetic contribution. But then when we look across locations, we're finding that it drops. Suddenly we have fall off. So the heritability is easy to explain within location, but actually when we look across locations, we actually find that it does less. It's not as good as explaining the phenotypes that we have. So we're back to this again. So we've done that earlier. So now all we're using here, we went from BLUPs and now we're using residuals because it was allowed us to have better normalization of the data. But at the top here, so we're going to talk about these in a moment. At the top here, we implemented I don't quite know how to say this because it's kind of an odd thing to say. I'm going to call it K-chair just because I was advised to say that. And it's basically an adjustment for population structure. And that was implemented by Alex Lipka, who's now University of Illinois, who's alumni from Cornell. And so we implemented the MLM using the K-chair population structure adjustment. And then we did that in just straight MLMM and then also in bootstraps of the phenotypes. We expect the bootstraps to be more liberal and the regular GWAS to be more stringent. So here we have QTL again, and we're doing liberal and conservative filtering for those. And we did that on the 83 million SNPs as well. Okay, so let's look at some of the data. So once again, what do we get? So this is for the bootstraps over here, then individual locations through here, and then looking just at the individual locations, whether two or more of those are observed in two or more of those are observed in each of the individual locations. And so for cadmium, phosphorus, and zinc, we have these numbers, but then looking at the total numbers across for all of the elements, we're getting these really big numbers again. But then we have 19 across all locations. So that tells us then that we're getting the same case again as we did previously in the NAM. But so we wanted to really look and say, well, how many did we get in the NAM and how many did we get in the diversity panel that are different? So what we have here is on the y-axis, we have all of the traits lined up there. On the x-axis, we're looking at the numbers of QTLs. And then the red is the diversity panel, numbers of diversity panel QTL. And then the blue ones are common to NAM and the diversity panel. And look how ridiculously small it is. Yeah? So the point is, but that's kind of what we expected. You know, we're looking at different populations. We're looking at different allelic diversities. We're looking at, in a way that we kind of should have expected that. Yeah? And so because we're looking at these different, like broader genetic diversity, we're actually mining a broader set of lines that we hadn't actually looked at in the NAM. So in a way, it actually confirms what we'd thought in the first place. So let's look at some of these genes again under these peaks that we were looking at. And we have iron, cadmium, manganese, and molybdenum. And then, do you remember way back in the beginning when I was talking about yellow stripe 1 when it goes into the root? Here again, we have yellow stripe 1. It's associated with iron. But we have these other ones that may be less, expected, but cutin biosynthesis associated with cadmium. But we can explain these away. Zinc transporter associated with manganese, maybe less expected. And then once again, our best friend, MOT1, has shown up again. So, but look at this. No, no, no. Three of those are actually not found in the NAM. But, our friend here, a qualitative friend over here, is actually found in the NAM as well. So we're finding that actually we're not finding them in the NAM, but we're actually finding them in the diversity panel as well. So the great thing with ZBrowse is that it allows us to really drill deep into the, zoom into the areas where we can actually see the genes. So this is our peak of different SNPs here, QTL, and here's a gene that it probably is associated with. And then also one for manganese, and we have this nice stack just sitting directly on top of this gene here as well. Okay, so I want to move on to the next population now, and this is the last one, so don't panic. So we're looking at the HIFs. And before that, I'm going to just sort of explain briefly how it is that we created the HIFs. So this probably looks familiar. This is the NAM recombinant inbred lines. And then from those, by design, we have a residual 3% heterozygosity across lines. Now, looking at QTL that we got from the NAM, we were able to then say, okay, so we're looking within this QTL, we want to actually find some heterozygosity that actually sits within that locus. And so what we did is that we looked within a locus, and then we're able to detect heterozygous regions. Then we select that one line that has that region, and then we plant it out. Well, we self-it, then plant it out. And then we'll do GBS, genotyping by sequencing, on each of these lines, and we're able to then select homozygotes for that region. So here we have 2 and 7, just random numbers that I picked here, that will actually allow us to find homozygous opposing alleles. So we take those 2 and 7 up here, and then we can see that they're different at that loci. And then we bulk those out and then disseminate them to the locations that we're working at. And here are some of the interesting phenotypes that we observed in Musgrave, if anybody's familiar with Musgrave. So that might be expected because of the interesting phenotypes associated with the elements that we're dealing with. Okay, so what do we do with these HIFs now? So we planted them in Florida, Indiana, Missouri, New York, Texas, 2 locations in CIMMYT and Puerto Rico. And the purpose of this, we expect that the HIFs will reduce the genetic variance that we see between lines. Also, we believe that it will increase the heritability of the traits that we're working with. And then also, we use the NAM-based QTL for selection of the HIFs, like I explained already. But then also, within each of these locations, we have areas of differences between the lines. So we really wanted to dig down into a greater resolution within that. So for that reason, we also used haplotype blocks as well. So this is kind of a zoom in of all of the lines. So this is a subset that you see here. So this is the haplotype blocks that we're looking at here. And each of these red lines that you see represent a haplotype block across the genome. These are the chromosomes. And then each of these boxes represents a QTL that we got from NAM when we did the NAM analysis. Now, the one that we want to look at right now, just as an example for you, is a cadmium QTL that we use to select lines. And so that's highlighted there in that section. So we really felt that the haplotype block analysis will allow us to get greater resolution within the QTLs that we're looking at for our selected lines. Excuse me. So once again, we go through this process of preparing the phenotype data. We're working now with 2.9 million SNPs for the genotype. And then now we're going to run a GWAS on blocks, the haplotype blocks, and then also GWAS on the individual SNPs as well. Now we're getting the QTL, and then we're going to be using the intersection of those QTL in order to find a QTL of high interest, a significant QTL. So this is for cadmium now. And on the left, oh, excuse me, on the left we have a QTL that is for blocks. And then on the right we have a SNP-based QTLs as well. So basically we're looking for the intersection of both of these. So here, using zBrowse, once again, we're able to align all of these intersecting QTL that we get from the blocks and the SNPs, and we have this one very clear peak. So when we zoom in on that peak, we find 2 subregions. So one is at 160 megabases, and the other is at 176 megabases. So we really wanted to know more about the details of this region. So we did a clusteral analysis on this. And here we can see that we have 3 clusters. So we had a trimodal distribution of concentrations. Now for cluster 1 over here, the green one, we found that we had a mean concentration of cadmium for those alleles of 4.82 milligrams a kilogram. So cluster 2, that's the black one over there, we had a mean of 27.78 milligrams a kilogram. So there's a difference of 23 milligrams a kilogram there. So that's a huge difference. So you can see the box plots at the bottom here for each of those clusters. So 2 and 1 here. So using a test of significance between these clusters, we're able to observe that there was actually a significant difference between 2 and 1, clusters 2 and 1 there. There wasn't a significant difference for cluster 3. So running the GWAS on individual locations, that was able to tell us that actually we did all of these locations at the top, but we only found those QTL in New York. So once again, that tells us that so many of our QTL are highly specific to specific locations. My voice is going. So. Okay. So that really gives us a greater idea about the G by E that we're working with. So what have we learned? We've looked at 3 populations here. So now we've learned, going back to some of the original questions that we were asking, what about biofortification? And here we're saying is if we really want to do biofortification breeding, we really need to understand the G by E and the application of that in QTL, using QTL that are applicable to specific locations. So GWAS helps us to understand the big picture of homeostasis. We can gain a knowledge of the complete set of genes. We've done that. We're seeing things. We're talking about diversity, and we get these QTL that come from that. But then also, we're getting location-specific QTL. We're getting a lot of them. So we identify QTL for locally adapted lines. And then we're also getting we can get strong multi-location QTL. But maybe we could have more success with those ones for planting across multiple locations in different regions. So for further investigation, so at the end of this, what do we actually get? What's the story that we can tell? For the NAM, out of 19 of 20 of the elements, we're getting more than 300 loci for each element. The one that's missing there is selenium. Sorry, Lily, I'm sorry about that. For the diversity panel, we're getting more than 250 loci per element, but that's for all elements. And then for the HIFs, we found the cadmium QTL and a possible modifier that was upstream at 176 megabases. So with that, I'd like to put things to a rest. And I'd like to thank my committee members. And who are they, you may be wondering. So that's Margaret Smith, Owen Hokanga, Ronnie's over there, and Lily. And sincerely, really, I might well up now, but sincerely for everything that you've done. I really, really appreciate it. Oh gosh, I am welling up. Okay. Anyway, and then I just want to say thank you for the funding that I've received as well from the plant breeding section. And then it feels like a huge amount of TA. I love TAing. No, TAing loves me, I think. So Steve Jesh in molecular biology and genetics, and then also working with Larry Smart and Michael Mazurik, and then also research funding from the NSF. And then at the top here, our collaborators, Ivan Baxter and Greg Ziegler, the guy that created ZBrowse. I think it's his last name, that's the Z bit in there, from the Danforth Center. And then also Chad Myers and Rob Schaefer. No, there's another project that I haven't actually talked to you about, and that was called a co-expression browser that Rob Schaefer was working on as part of his PhD. And what that was about, so you know how we've actually drilled down to the gene level, and we're kind of interested, well, what's beyond that? How does that fit into a genetic pathway? What's the molecular sort of mechanisms behind that? And really, as things are, as we look at them at the moment, we're unable to do that. We can guess, but we really want to sort of drill down even further into understanding. So that's something that Rob was working on. But in the timeline that I had in order to finish up, it didn't work out. So anyway, so we also want to thank people for germplasm and the planting locations that we use. So in Texas, Seth Murray, Natalia Palacios, and Aldo Rosales in CIMMYT, Jeff Gustin in Florida, Sherry Flint Garcia in Missouri. And then here at home, Cornell University, Sherry Norman, Keith Payne, and Dan Fisher on the Corn project. Also want to thank Denise Akdemir. As time was running out towards the end, it was like, I've got to do this, I need some help. So he was able to help me from the statistical consultancy unit. But most, but not least, where are they? Oh gosh. I'd like to thank my beautiful wife and my beautiful daughters. They're actually sitting at the back here today. Can you stand up? So my wife, she's supported me through thick and thin. Thank you. Okay. Anyway, I didn't think I'd be so emotional. So with that, I'd like to, any questions? Yes. Yeah, please.
So, just to be clear, we'd like to thank the Maize Diversity Project for growing the NAM for us. We didn't have to grow the 5,000 line plus checks in 4 different sites. We're very grateful that Ivan, Chad, Rob, and I all got to go out to Missouri to work with Sherry to package said corn so that we could start doing chemistry almost immediately. That was a huge advantage. And then also thanks to other people that grew the 282, like Rochford in Indiana, and Jonathan Lynch, who grew it in New Zealand, South Africa. Thank you, Owen. Sure. Many people to thank on this project. So, questions, comments for Philip?
I prefer comments. Yes.
The nitty-gritty, but what are the exact criteria for deciding whether 2 QTL are the same across 2 locations?
Right. Yes, sorry, I was meant to say that. So thank you. Thanks you for raising that up. So basically, when we were looking at the NAM, the diversity panel, very often we didn't have SNP for SNP exactness. So basically what we did is we created a 2 KB window, either side of the locus that we were looking at. And anything that fell within that, we considered it to be the same, assuming that, you know, looking at Jason Wallace's recent paper, looking about how within a 20 KB window upstream or 10 KB upstream and downstream, we felt that would be a reasonably conservative method of doing that. Yes.
Just following up on that question, do you see a very small subset of QTL being able to replicate across multiple locations? Have you tried to compare, say, what qualifies a multi-location QTL versus the vast majority that are location specific? Because that might tell you something that's not logically relevant about what those QTL are actually doing.
Right. Yeah, right. So at the moment, yeah, so absolutely. So that's something we'll need to look at. But I mean, based on our multi-location QTL here at the moment, it's all it is the QTL that we can actually find and detect within one location, another location. If they're exact hits, then we can call them the same thing. But that's something we'll need to look into at some point, yeah. I can see you're busting to ask me questions. But yes.
I think you sort of, to state, you partition as genetic and environmental, sort of the factor, G cross E.
Yeah.
Could you also say that nature, nurture, phylogeny, ontogeny?
You want to talk to me later?
I'm a rank amateur. I'm just.
No, it's fine. I suppose you could say that, maybe, yeah. G by, yeah, nature, nurture. That might be a way of putting it, yes. So the nature would be, you know, what's the genetics? And then the nurture would be the environment that you've grown up in. So basically, that's exactly right, yeah. Does that help?
Thank you.
Oh, okay, yeah. Yes.
How many kernels do you use for each plot?
How many kernels?
And do you think it would be valuable increasing the sample size to increase it?
So regarding your last question, the sample size, so that's not something that I was actually involved in. That was something that Greg Ziegler was doing in the Danforth Center. I believe that they took something that was a happy medium. Am I in the right?
So there's the experiment you want to do, an experiment that you can get us have to actually pay for. Fortunately, from the NAM design, the B72 check appears more than 150 times. So that's probably, I think we shot for 5 hurdles per packet. So that's very well replicated. If you actually look at what was sampled in any given site, it's not 5,000 volts. It's probably closer to 3,000 volts. Because of who didn't germinate, who was fit to us, whatever. So, yeah, deeper would have been better. But actually Baxter did power analysis while we were iterating the grant as to what was the acceptable amount of sampling to get started. And so you can actually look at that.
Well, we're about out of time, but thank you for coming. Thank you for your attention.
This has been a production of Cornell University, on the web at cornell.edu.